So my lab studies a mutualistic association of plants, plant roots and certain species of soil fungi. You can see a picture of one down here on the left. They belong to the subphylum Glomeromycotina. And these two organisms have associated for a very long time. So the ability to form these symbioses, these mutualistic associations, arose very early in the plant lineage, 400 million years ago. And so if an association has been maintained for that long, which it has in the majority of the flowering plants, then you might expect some particular benefit for the plant. And probably the most well-described benefit is that the fungus assists the plant with the acquisition of mineral nutrients from the soil, particularly phosphorus as phosphate, but also some nitrogen. And the plant supports its symbiont with carbon. The fungus is an obligate symbiont and is entirely dependent on the plant for a source of carbon. And they associate in the plant roots. You can see up here on the couple of the images, the fungus is inside the roots, inside the root cells. Those darkly staining blocks that you can see on the left panel are the fungus inside the root. And it forms inside these cells very branched structures, hyphal structures called arbuscules. And in order to accommodate the fungus, the plant cell needs to really rearrange the whole of its cell biology, its physiology, metabolism, in order to accommodate this huge endosymbiont in its roots. One of the major changes in the cell includes building a membrane that surrounds all of those hyphal branches so that the fungus actually resides in a new membrane-bound apoplastic compartment inside the cell. And this is where they exchange nutrients. So the phosphate that the fungus captures with external hyphae is transferred to the plant at this point, and the plant feeds its symbiont with lipid, again, across this interface. So the main questions that we're interested in, I think, are how does this association develop and how does it function? And because the plant is the tractable partner, genetically tractable partner, we really look at it through the lens of the plant. We're asking, what is the plant's genetic program for allowing symbiosis? How does it change the biology of its cell to accommodate this organism? And the approaches we use, I would say, are really grounded in genetics. So we start off by looking for plant genes that are important for allowing the fungus to be accommodated inside the cell. This can be through forward genetic screens or reverse genetic screens. There's an example here of a mutant plant on the right in which the fungus is unable to be maintained. It turned out to be an ABC transporter that exports the lipid to the fungus. And once we've identified an important gene, the reason we start really with genetics is to find something that is really central for the symbiosis. Then we would do all sorts of different things to characterize that gene and put it into the context of the biology of the cell. Sometimes that's cell biology, sometimes it's biochemistry. In the case of the transporters, it might be transport biology. So a lot of different things go on. I think for us, technology has always brought new opportunities, and particularly new technologies and collaborations have allowed us to make many of the advances that have gone on in the lab. Just to give an example, some years ago, when sequencing became very cheap and many labs were sequencing genomes and genomes became available, we were able to use that information to make comparisons very broadly across the plant kingdom with a phylogenomics kind of approach to ask what genes exist in plant AM hosts, symbiosis hosts, and not in non-hosts? And would those be particularly important for symbiosis? And this was a really nice collaboration with Lucas Mueller's group and allowed us to home in on a small set of genes in Medicago truncatula, 138 genes, that are really, now that we've studied them, are really adaptive for allowing the plant to form and function with its symbiont. And so the new technologies that we're interested in at the moment are the new transcriptome, highly resolved transcriptome technologies that allow you to look at the transcriptome of every single cell potentially in the root at any given time. And so there are different ways to do this, single nuclei sequencing or spatial transcriptomics. And so what this gives you, well, what we've done before was look one gene at a time through promoter Gus fusions to see where a gene is expressed. This is on the left panel. But what the new technology allows you to do is really have that information for every single gene in the genome across the root. So you can do this spatially with different kinds of platforms. You can also do nuclei sequencing and then put those back into groups with markers and a lot of technology. And this is being spearheaded in the lab by Trevor Tivy. We have a collaboration with Ewan Vamick's lab in biomedical engineering. They're experts in this. And if you'd like to hear more about that, Trevor will talk in the atrium in the next session. Thanks.